All right, David, welcome to the podcast. How are you doing?
Yeah, no, great. Good to be here. Yep.
It's a pleasure to have you on. For the listeners who don't know who you are yet, can you tell us a little bit about who you are and what you do?
Yeah, I guess like a lot of people in my field, I started out more in sports science. Actually started out as a PE teacher, then went back, did research, sports science, and worked with sporting teams and institutes of sport for a little while. And then I guess it gradually transitioned more into a research focus. And still got the passion for sport and improving performance, but like a lot of people, as I get older, also getting a bit more interested in how some of the training principles can be applied to improve health as well.
Hmm.
And I'd probably say that, yeah, I think that's where one of my main interests is now, I guess, is, yeah, athletes, have such, well structured training programs, whereas sometimes exercise for health is kind of maybe not as prescribed, and we maybe don't want to get as prescribed, but I think there's a lot of scope in using yeah, how do we prescribe exercise for athletes? Can some of those principles also be used to better improve health?
I guess that reminds me of, are you familiar with Inigo San Milan? I'm sure you
Yeah,
know
yeah,
the,
of course. Yeah.
yeah. It sounds very much like his philosophy around this, where he works with elite athletes, elite cyclists, and looks at, how metabolism can be developed in those athletes, and then tries to understand, well, now this is quote unquote the best stuff we can make. So how can we then transfer that over to the health side and try to make people that are unwell or sick? How do we get them towards that optimal frame? Do you recognize yourself a little bit in that framework?
Yeah, and it's interesting because you sometimes like you don't want to be sort of too evasive in your answers, but quite often, and as with athletes, you kind of get asked, "Oh, what's the best exercise?" Like this idea that there's one magical exercise that's going to do everything for everyone. And and when you think about athlete training programs, obviously, well, I don't think too many elite athletes go out and do exactly the same, training program every day of the week, every day of the year. And so I think trying to get a little bit of that into the health field, that there's times when you might want to do some high intensity, there's times when you're doing low intensity. You need to be able to hit different aspects to get the different health benefits. And I sometimes also refer to the classic sort of diet pyramid. And I think it's the same with exercise. To draw that analogy, you're not going to say, look, just go and eat this food or whatever. And I think it's the same with exercise. There's lots of different ways and intensities and volumes that you can exercise. And I think that's one of the things that does excite me is, I don't think we'll ever get to the perfect, but how do you, based on individuals, how do you put together a training program that hits a lot of the different aspects that you need to lead a healthy life and to have better performance?
Yeah, and I definitely want to get to that and get your point of view on how we can best organize those training areas. And I guess to respond to what you just said, it's more a question of and than a question of or. And in today's world, everybody's about the or, and we have to pick between one side and the other. When usually we need both together. It's just a matter of how do we blend them together.
Yeah, it's interesting. I think a lot of smarter people than me have had this discussion, but we do in some ways seem to be, there's like a little bit of a binary thing happening in lots of aspects of life. And I think that can, nutrition or exercise, that can pop in. I've seen, when you're on the internet and different things like that, like this is the exercise that is the one that you should be doing, and this is the one you shouldn't be doing. And yeah, I mean, sometimes those simple messages are needed, but obviously, as you know, and probably people listening know, it's always a little bit more complicated than that.
Absolutely. So before we get into the macro perspective, the big picture and training plans and stuff like that, I want to go back to the micro. And one of the things that you've been studying for many years is, amongst other things, is mitochondria. And it's an organelle that absolutely fascinates me. So I wanted to maybe start at the beginning. What are you most interested about when it comes to the origin itself of mitochondria and the symbiotic relationship that we have today with that entity, let's call it?
Yeah, like I said, I think I'm not even sure how I got into that field, but I think, yeah, there's just something cool about mitochondria. I mean, obviously, just being the powerhouses of the cell. So the fact that you're getting 95% of your energy suggests that they're pretty important. But even aesthetically, they're pretty cool to look at. The mitochondria when you see we do some of the microscopic work. So I mean, obviously, most people have seen drawings of them, but then when we've seen them under the microscope, I think they're just a cool organism. And like you said, I think if when people go back more into the who are interested, the fact that somewhere along the line the bacteria and the mitochondria have merged and gradually still a little bit interlinked, like they haven't completely separated, so they've still got that I guess symbiotic relationship where the cell depends on the mitochondria, and the mitochondria also depends on the cell to generate new mitochondrial components. So it's an interesting organelle in that respect as well.
You talked about its structure, and we often see it drawn out as almost like a bean-shaped organelle. Something I've seen recently is those images of the mitochondrial reticulum, which is really a network and an infrastructure within the cell itself. Can you talk to this point a little bit?
Yeah, it's like lots of things, and I guess the way I kind of think about it sometimes is that if you go into your delicatessen or whatever and you get a sauce, if you cut through that, it's basically going to look like a circle. And so that's what you're mostly seeing, in the textbooks and things like that, because what people do, get a piece of muscle, and you, not unlike what you're doing in your delicatessen, but with a much smaller piece of meat, you're slicing through it. So for a lot of time throughout history, we've only had like a two-dimensional image of what the mitochondria looks like. And as you said, what we know now, that it's not just these sort of isolated spheres floating around in the muscle, but it's more these tubular works that are interconnected. And there's we haven't done this really cool live imaging that the mitochondria are also dynamic. So when you're exercising, when there's energy needs in different parts of the cell, you see like these little worm-like structures almost, moving through the muscle. So you're taking these energy powerhouses to the places within the cell where they're actually needed. And when you think about it, the mitochondria are, depending on obviously the level of the athlete, but take up around about 5% of the volume of a cell. So 95% of the cell doesn't have mitochondria, but there are going to be energy producing or energy using processes going on in all those parts of the cell. So it appears that mitochondria are not just these static, spheres fixed in the muscle, but these dynamic tubes that can grow and move through the muscle and go to where the energy is needed.
That's quite fascinating. I didn't know that they could actually move and be so dynamic. We'll go into the power aspect of the power generating aspect or recycling aspect of the mitochondria. What other roles does the mitochondria play in the human body or in a human cell that maybe people don't know about?
And I think because they are it's a classic when you read a science article or something sort of for the general audience, they'll talk about the powerhouses. So that's what they're most well known for. But they're also involved in generating reactive oxygen species. So a lot of people be familiar with that aspect as well. They're also involved in not only just linked to the health, but in signaling functions, but also linked to different health functions as well. And if you look through the literature, a lot of the diseases, I guess, that mitochondria function and content has been linked to some of them make complete sense, like things to do with substrate metabolism. So you might think, diabetes and obesity, that makes sense. But a lot of neurological diseases as well, so Parkinson's and Alzheimer's and things like that. So they, as well as producing energy, they've got this pivotal role in quite a range of different conditions or diseases.
My understanding is that red blood cells are one of the only parts of the body where there is no mitochondria. Is that correct?
Yeah, and there's been so in the actual red blood cells itself, you're right. There has been some what they call, and this is a pretty maybe getting a little bit off topic and a bit of debate at the moment, but there has been some studies showing what they call cell-free mitochondria, so mitochondria floating around in the blood, which actually seem to be functional. So that's kind of a bit of a debate at the moment. But in terms of the red blood cells, you're right.
Do we know why this is one of the only cells that doesn't have mitochondria?
No, maybe someone does, but yeah, I I haven't come across, and most of my work is confined to the muscle, but yeah, I think as you're alluding to there, I mean, basically you find mitochondria everywhere throughout the body, in different, in the brain and in all different types of cells, but why the red blood cells themselves don't, I'm not sure.
Can you give us a bit of a rundown on, let's focus specifically on the muscle cells themselves. If we think about different muscle fiber types, what I've learned through textbooks and other informational content is that there is a significant difference between mitochondrial content in different fiber types. So what's your perspective on that?
So we're talking about just for, I think most people are probably aware that within the muscle there's different fiber types. So you have in humans at least, you've got type 1, which is the slow twitch fibers, and they'll be recruited at lower intensities. And then as you increase the intensity, you'll increasingly recruit the type 2 fibers. And we have type 2A and type 2X, and they'll get more recruited as you start going, faster, higher intensities up to sprints. And the type 1 fibers, because they're more involved in endurance type activities, they tend to have more mitochondria than the more glycolytic or fast twitch fibers.
Okay, and is there a big difference in the development potential of the mitochondrial pool in those different fiber types? For example, does the type 1 fibers have a bigger potential for developing that mitochondrial content relative to the type 2 fibers?
Yeah, I haven't seen any evidence of that. I guess maybe to partially answer your question is that what you will see, especially if doing a lot of endurance training, the type 2A fibers will become more similar to type 1 fibers. So that difference will get reduced with a lot of endurance training. So I think that probably, I hadn't thought about in exactly how you asked that question, but I think that would support what you're saying there. So when you do more endurance training, you'll get greater increases in mitochondrial content in the 2A fibers, such that they start to look more similar to the type 1 fibers.
One thing I've come across, thanks to your work, is maybe the distinction we can make between developing mitochondrial volume density, as you call it, and mitochondrial function. So could you explain those two, let's say, axes of development of the mitochondria, and then we can get to what type of training is going to affect which part of it?
Yeah, no, it's a really good question, and I think for people who are reading the literature, sometimes if you're going to scientific papers, sometimes it can be a little bit confusing because those two terms will almost be used interchangeably. And I've seen sometimes people will talk about increasing function, but they're increasing content. I've been racking my brain trying to have a good analogy, so I haven't come up with a good one yet, but basically, so the first one is the content or the volume density of mitochondria and this isn't exactly the right term, but you can think of that in terms of the number of mitochondria that you have.
Mm-hmm.
And as you said, you've got the reticulum and they're joined. So number isn't the best thing, but I think to understand this concept, it's good enough to be able to understand it more clearly. So that's really how much of your muscle is made up by mitochondria. And in most people, who are untrained, not doing too much, it might be about 3 or 4% of your muscle is made up of mitochondria. And in really highly trained endurance athletes, and we've seen some stuff in cross-country skiers, they can get up to 8, 9, 10% of the muscle is mitochondria. So that's how much mitochondria is in the muscle. Separately to that is how efficient does that mitochondria use oxygen to convert that into ATP? So you can think of it in terms of, if you doubled the amount of mitochondria you've got in your muscle, you should be able to then also double the amount of energy that your mitochondria are capable of producing. But there's also, depending on type of training, you can improve what we call the quality of that muscle. So you might see an example where you double the number of mitochondria, the amount of mitochondria, you triple the amount of ATP that mitochondria can produce. So that's when we're talking about the mitochondrial quality. And so we normally talk about in two ways. One is how much energy can all of your muscle produce, and so that depends on energy can you produce per mitochondria. So that gives us a measure of the efficiency of each individual mitochondria. And as you alluded to at the beginning, we've seen all those different combinations where you can increase content and you can increase the at the same time, you'll increase the amount of total amount of energy you can produce, but we can also not increase content and increase the amount of energy that the mitochondria are producing.
Okay, and I've also heard the terms mitochondrial fusion and fission. Where do those fall on that spectrum?
Yeah, so slightly different. So I guess like a lot of things, I guess a mitochondria has its own little life cycle. So fusion is exactly what it sounds like, two things fusing together. So when you, especially in endurance, with that type of high volume and endurance training, you'll get these separate mitochondria fusing together and creating longer reticulum.
Mm-hmm.
And so that's the mitochondrial fusion process. But also what's equally happening over time is, I think if you can, imagine if the mitochondria is getting, an individual mitochondria is getting either damaged, or maybe part of it is coming towards its end of its life cycle, it'd be inefficient to destroy all of that whole mitochondria. So what you'll also do is you'll get sort of like this pinching off. And so as the opposite, basically the fission is kind of like the opposite of fusion. So instead of making longer mitochondria, you're then budding off a smaller portion of the mitochondria, and that'll typically then be engulfed by a lysosome, and that'll be destroyed and the contents sort of recycled over time. So you're constantly going through this process where you've got a mitochondria, you've got them fusing to make longer reticulum, and in that life cycle, then you've also got, so that's the fusion, you're also going to have fission happening, where you're then making smaller mitochondria. And if any of those smaller mitochondria have got some sort of damage, then that part will be destroyed. And as I said, the components will be reused, and you'll keep going in this circle of life for the mitochondria.
So when we aim at, let's take the first one, increasing, like you said, the number of mitochondria or the volume density of the mitochondria, what is going to happen to the mitochondria itself, and how is it going to essentially grow in numbers or grow in volume and space?
Yeah, so the mitochondria, I guess, are fundamentally made of two things. They're going to be proteins, and then you've got lipids in the membrane of the mitochondria. And like other cells, the proteins will sit in that lipid membrane. So, for example, when you're doing exercise, you're stressing the body, you're stressing the mitochondria, and so that's going to be a stimulus to increase more mitochondria. So there'll be a synthesis of those proteins and lipids, and they'll be incorporated to increase the length of the mitochondrial reticulum, and also will probably activate at the same time fusion to join different mitochondria together. So one of the things that I guess is maybe different to how people might think of it conceptually is you don't get these new, like you don't get a new mitochondria appearing like from scratch, like a little baby mitochondria growing into an adult mitochondria, but you start off with a mitochondria and you add to the mitochondria that's almost there. So almost like a house extension or something like that. So you're not building a new house, you're adding a room or whatever onto that mitochondria. So those additional proteins and lipids will be incorporated to increase the size of the mitochondria that are already there, rather than, like I said, you don't have a process where these little baby mitochondria are born and grow into new mitochondria.
Yeah, that's really interesting to think about. And is it the same when we talk about mitochondrial function or respiration? Is the process similar or is it different?
I think you've hit on a really great question there. And I think the simple answer is we don't really know. And so, we've done lots of experiments. And so part of the increase in the improvement in the ability of the muscle to supply energy that you'll need, for endurance athletes or whatever, is to do with having more mitochondria. And so that's easy to explain. You've got more mitochondria, you've got more of the mitochondrial complexes that can take oxygen and ATP. But as I said, we've also seen with different types of exercise where there's a disproportional increase. So the mitochondria seem to get better at producing energy somehow. And we don't really know, what's behind that. What change is happening that's allowing the mitochondria to be, per amount of mitochondria, being able to produce more energy.
One maybe a beginning of an answer that I think I've seen in one of your webinars on training in mitochondria was the idea that, especially if we talk about very high intensity training or high intensity training, a part of what was going on was essentially the killing off maybe, or the recycling of older mitochondria that didn't breathe very well, that didn't respirate very well. And then in place of those older components, the body would just, or the cell would make new ones. And those new ones seem to breathe better. Is that correct? Or is that back on the table and in question now?
Yeah, I don't know about correct, but that's one of the theories that we had. So I think you've summarized it really well in that, as I said, so mitochondria are constantly going through this life cycle where they're growing, and then as they I guess come to the end of their life cycle, then parts of them will be budded off and broken down, and then the bits will be recycled to produce new mitochondria. So that's happening all the time. And I think, it's probably, it's analogous to what's happening to your, I guess, skeletal muscle as well. And I don't know if you've had some people on to talk about this, but I think basically sort of every 6 to 9 months, you've got all the proteins in your arm, for example, are completely new.
Mhm.
because you've constantly got the protein being broken down and resynthesized. So yeah, I think that same thing is happening in the mitochondria, and with resistance training, you're kind of accelerating that process as well. But what we have seen is that, especially with higher intensity exercise, we've seen some damaged mitochondria. And what we suspect that you alluded to there, what we think is that because we don't see changes in mitochondrial content, even though there's more mitochondria that are damaged and going to be marked for breakdown, is we think that that may be speeding up that process of moving damaged or mitochondria that aren't functioning as well, moving them more quickly to the breakdown process so they can then be, the components recycled and made newer, better mitochondria.
Okay, that makes sense. Um, Another aspect of mitochondria that's really interesting is that it has its own DNA. Can you talk a little bit more about that?
And so we touched on that right at the beginning because of once upon a time, mitochondria would have had their own, made proteins, but over time, because of the fusing with the cell, most of that responsibility has been given over to the cell and the nucleus in the cell. But so I guess it highlights the complexity is that we still don't know how many proteins there are in mitochondria. We know that there's a lot, and somewhere between probably 1,200 and 1,600 proteins are actually in the mitochondria. The vast majority of those is exactly what happens everywhere else. So you have the cell signaling that's activated, and they'll be synthesized from outside the mitochondria, and then those proteins will be imported into the mitochondria. But for some reason, and probably a remnant of, as I said, the origins of mitochondria, I think I always get this number wrong, but I think there's 13 essential proteins that are derived from mitochondrial DNA. And so those proteins are essential for mitochondrial function. But for some reason, because of as I said, the origins of mitochondria, those 13 proteins are still synthesized, are still produced from mitochondrial DNA. So you've got a unique situation there where the final mitochondria is a combination of nearly all the proteins being synthesized outside and imported in, but a really small fraction. So if you think about, if we round numbered and make our calculations easy, but 13 out of about 1,300 proteins that are still synthesized from mitochondrial DNA inside the mitochondria.
And that leads into my next question, because we know that we inherit our mitochondria or mitochondrial DNA from our mother almost exclusively. So does that mean, for going back to what you just said, 13 out of 1,300? So actually, one of the questions that I had was, can you inherit, let's say, poor quality mitochondria from your mother, and is that going to have an impact on the quality of your life or how much you can develop that mitochondria? But according to what you just said, maybe given how small the fraction of proteins that are coming from the mitochondria itself, maybe that's not as important as I thought it would be.
Yeah, it's a really good point. And I think it's interesting because sometimes, not just because of the numbers, maybe not so, but some of those proteins are really important because they have, really important roles in respiration. And so there are, well, exactly you said, so mitochondrial diseases that are inherited from the mother. I mean, that's how they will be inherited. And again, this is not my area, and I'm not sure what the situation is in the US, but I think in the UK and in Australia, they've they've there's a they've changed only recently changed the law so that there are some clever scientists that when they can do IVF, but remove just the mitochondrial DNA part.
Okay.
you're getting all the other inherited information from your mother, except for the mitochondrial DNA. So that gets replaced to prevent the mother passing on some of these mitochondrial diseases. So you're right that I said like 13 out of 1,300 or so is a really small fraction, but it can still be important. And most of those, because I guess of how important the mitochondria are, most of those mitochondrial diseases are, quite debilitating, and also a lot of children will die quite young if they do have a mitochondrial disease. So as I said, there's now increasingly, and some, I guess, depending on, different countries, and because of, thoughts around genetic manipulation, can be a little bit scary for some people, but there is a process to be able to remove just the mitochondrial DNA that's from the mother and do IVF to try and prevent the transmission of some of those diseases.
And before I go any further, I want to give a shout out to my friend Vincent, who sent me some great questions to ask you regarding mitochondria. He's been digging into the topic, and that was one of them. And maybe pushing that a little bit farther, then, if we put aside mitochondrial diseases per se, is there a high variation of quote unquote quality of the mitochondria from one person to the next that is inherited? And does that impact whether it's the lifespan, the quality of life, the potential of development at that level? Or essentially, as long as you don't have any mitochondrial disease per se, pretty much everybody's playing or is on a level playing field in that regard?
Yeah, again, thanks, Vincent. Tough question. I haven't looked into it, but I think there is still, like with most aspects, there's a genetic component to, all of our physiology. So I mean, there's some people who naturally have bigger muscles who are stronger, and others that are better at endurance events and things like that. So, and obviously the mitochondria is one part of it, but you definitely see, for example, following on from our discussion earlier, people will have born with more slow twitch fibers or more fast twitch fibers, and you will also see people who are born with more mitochondria and less mitochondria. In terms of, we're talking about the kind of the content versus quality debate. I haven't actually seen, there's probably something out there and I haven't looked into it, but I haven't seen any evidence that there's differences in mitochondrial quality, but definitely inherited differences in mitochondrial content.
Okay. Moving to the training questions, and then we'll circle back. I mean, they all blend together, but I also want to talk about the health aspect a bit more in detail afterwards. There has been a few great papers and discussions going back and forth on the debate of high volume versus high intensity. Could you summarize a little bit your position and the opposing view on that question as well, and then we can go into more detail on that?
Yeah, and let me just start by, I think just the terminology is always important. So I think there's not always a big difference, but when we do volume, we're talking about, I guess, the total amount of exercise. So it's not just the time. And so I guess an analogy in terms of, like strength training would be if you took the number of repetitions times the number of sets times the weight.
Mm-hmm.
That would give you like a total volume of weight lifted. Mm-hmm. So when we calculate, when we talk about this in terms of volume, we basically take the intensity times the number of minutes that you're doing the exercise. So so if you did, to give like an example, like if you did 30 minutes at low intensity, that would be a lower volume than 30 minutes at a higher intensity.
Mm-hmm.
So just because sometimes
could we equate that to work then?
Yeah, exactly. That's what we reported in kilojoules.
Okay, yeah.
So it works. So sometimes I think, because sometimes, I've seen in terms of training, people might say, oh, the volume of training that I've done is this amount of kilometers or this many minutes. So
Hmm.
just so it's not confusing people who might be using that term.
Hmm.
And so what we've seen, and I guess, again, it's not a binary thing. So we've seen that high intensity exercise and even sprint interval training can increase the mitochondrial volume. But what we've seen is that our research at least is that the changes in mitochondrial volume, or the amount of mitochondria, correlate pretty well with the volume of training done. So the more work you do, the greater the improvements, up to a certain point in the content of mitochondria. So it's not to say that high intensity exercise can't increase the mitochondrial content, but we see it proportional to the total amount of work that's done to training. If you wanted to take and I think sometimes, we did this debate with Marty Gabala. I'm not really sure if there was a winner, but if you wanted to do it in terms of time, you could maybe make the argument that, really high intensity training is more efficient because you can do it in less time, but you can't get a large amount of work done if you're doing sprint interval training. So I don't know if hopefully I've answered the question there, but fundamentally I said that we've seen a pretty good relationship between the total amount of work done with aerobic training and the changes in mitochondrial content. So we think that the amount of work that you do is the main determinant of the changes in mitochondrial content.
Mm-hmm. And one thing that I think is useful in that conversation as well is to think of the cost of those different modalities and the way that you train. And like you said, sprint interval training, for those who have done that, they're all out sprints, and I think technically it's anywhere between 6 and 30 seconds all out. And for anybody who's done a Wingate sprint, they'll know that it's no joke. And my argument is always that you can do a study for 6 weeks or 8 weeks and get people to do those really, really hard efforts 2-3 times a week. But the higher level you are, the more taxing those efforts are going to be, and so the less you can technically do and assimilate. Would you think along those lines as well?
Yeah, and I think like you say, I mean, there's just no way you can get the volume that's required there. And when we do our studies, we always make sure we've got a bucket on standby as well, because those sprint efforts are that if they're done all out, they are intense. So, and we'll maybe get to that later. Like we started off by talking about having different types, different components to your training. So I strongly believe that there is a role for sprint interval training. But if your goal is to try and maximize your mitochondrial content, I think there's no escaping that you need to do a high volume of training.
And like you said, we'll come back to that because I do believe that the answer lies somewhere with the and not the or. When it comes to developing that to the utmost degree, that mitochondrial content, we can assume that it will be highly beneficial for longer duration events and sports. We can think of endurance sports. Is there a good argument for non-endurance sport athletes to also try and push up their volume of training and develop that mitochondrial base?
Yeah, it's a good question. I think it's hard to have a definitive answer because I think there's always so much you can do in a week. So if you think about a team sport athlete or whatever, you want to be strong, you want to be fast, and you want to be able to recover. Between the efforts, and obviously that also depends on the nature of the sport. And so I think when it comes to a coach, they're going to look at the weaknesses and not trying to improve everything to the max. You know, there might, for whatever reason, you know, the focus might be strength, power, sprint speed, as well. So I think there still is value, and you know, we've seen some of the work we've done more with repeated sprints and things like that. I think the mitochondria, the if you think about like the recovery, so when you do a sprint, you're breaking down ATP and phosphocreatine to give you the rapid energy that you need, and then you need to be able to resynthesize the phosphocreatine to be able to do subsequent sprints, and that's an aerobic process that is going to depend on probably the amount of mitochondria that you have. So I think there is some value in that volume of training to be able to improve recovery between sprint efforts. But you know, if you're a slow team sport athlete, I'd be focusing on the power and, you know, the speed first. So I think that's another, you know, that's one where I'm not trying to be evasive, but yeah, it depends on not only the requirements of the sport, but also where the individual athlete sits in terms of their particular strength and weaknesses in those different capacities.
Yeah, and to your point, I have a study in mind that was done on soccer players, and they looked at repeat sprint ability, and they built a model, and that was highly predictive of the results. And the two parameters that they included was max velocity, so the top speed that they could hit on their sprint, and the second one was the speed at threshold 2, if we want to call it that. And I guess there's going to be a high relationship or a strong relationship between the amount of mitochondria that you've developed and how high that second threshold, FTP, whatever we want to call it, is. And that seems to have a big impact on someone's ability to repeat sprints, recover fast from those high intensity efforts, and then be ready to go again.
Yeah, no, I think that's spot on, and we've seen very similar, and we haven't published this, but I think some others have done some similar stuff, but we've seen like a really strong correlation between the second lactate threshold and mitochondrial respiratory function as well. So I think, yeah, you basically hit on it there. I mean, we've seen like you get to be good at repeated sprints, you need to be fast on the first one, and you need to be able to recover as best as you can. So I'd say that you're spot on with those two key physiological capacities that need to be developed. And I think what's interesting as well, I was ages ago, and we haven't published this either, but you know, and I was actually just having this discussion a couple of days ago, but with a lot of the team sport, we've tested some team sport athletes and really good VO2 maxes as you'd expect, but lactate threshold was just terrible. Like basically they just start producing lactate straight away. And some of them, if you can, talking, so it's a bit hard visually, but remember some of our best athletes, and this is like a Premier League football in Australia, but they almost had like a 45-degree line from the first, you know, there was almost no, I don't think there was a threshold at the beginning of the season. And I think sometimes it's maybe a little bit, not controversial, but also counter what people would think. But you know, team sport athletes can do a lot of really high intensity sprint training, which is obviously important. But I think sometimes what they're sacrificing is some of that lower intensity, high volume, which is important to push up that second lactate threshold.
Yeah, and for a bit of context, because we haven't spoken before, I work a lot in CrossFit and other high intensity sports, and I see the same profile where they're able to output crazy amounts of work, especially at high intensity. But then when you test the engine itself and how it functions, there's a lot of room for improvements, and it usually doesn't require, you know, hundreds of hours. I mean, it is going to over the long run, but sometimes as low as 2 hours a week of low intensity training can help those people that, again, already have a very good athletic level, strength level, power level, just potentiate it better maybe. And like you said, recover. And that's one of the things that I see the most is people just recover better and can do more work as a result of that. And if, and I mean, we know it's not it's not a causal relationship, but there is a strong correlation between total volume of work and then performance in competition. And that's true across the spectrum of sports, whether we look at Olympic weightlifting or biathlon on the other side. Those are going to be influenced to a high degree by total amount of work. Do you see that I guess that mitochondrial capacity, if we go from the macro to the micro, as being one of the components of being able to sustain high workloads and recover from them and adapt and keep progressing?
Yeah, absolutely. And I think, I mean, I agree exactly with what you're saying then. I think it's part of playing the long game, is that by doing some of that lower intensity, you improve your recovery, which will improve your ability to do the high intensity work when you need to do it. And I think it's interesting. It's a really interesting argument. I've done a few papers on this, but I think sometimes, training can be a little bit too focused on the specific, like doing training that's aligned with the actual event rather than improving the physiology that's important to do the event. And so I sometimes, I don't want to criticize anyone who's listening, but I think sometimes too much effort is spent trying to replicate the sport in the gym or in the training, and the same intensities, whereas I think that's not always going to give you the physiological gains that you need to be able to perform at your maximum, if that makes sense.
Yeah, to your point, what I've seen, especially if we look at what the best do and try to, and obviously we cannot just replicate what the Olympic gold medalists do and think it's going to work for everybody. But one clear pattern is obviously the difference in how their training looks from 1 month to the next, or at one part, maybe preseason versus in competition season. And it seems like the people that do best have a high variance between those different phases in their training, whereas people that maybe can operate at a good level but seem to reach a plateau, their training might look very similar all year round, whereas for the others, you have those distinct phases, and like you said, maybe working more, and we go back to very, I guess, basic principles of periodization, of having general training and then having specific training and then competition, and sequencing those blocks in whichever way it makes sense, obviously depending on your sport. If you're a cyclist or in an endurance sport, your season's going to look very different than a rugby player who has to, be in top shape, arguably every weekend, most weekends of the year. But I think you hit on a good point there, the specific versus general. And it does seem like a lot of time is spent thinking about the specific and maybe not enough about the general.
Yeah, and then that's when I sometimes, depending if I'm giving these talks to coaches or athletes, that's the way I like to think about it. What's the physiology that I need to do my sport at the highest level? And then, okay, how do I design my training to improve the physiology that I need? And obviously there's other things, there's also psychological and technique and all that sort of stuff. But I think, to me, that's probably a better approach. And I know also I've seen with athletes as well, that sometimes when you've got a group of athletes, the tendency can be to try and, for maybe some of the lower level athletes, to try and train at the same intensity as the best athletes in the group. And you can do that initially, but what I've often seen is that, kind of like this wedge growing, that as the season goes along, the slightly lower level that trying to keep up with the better athletes, they don't improve as much as the athletes who are hitting their training zones and training in their appropriate training zones and having that big difference between their easy, their medium, and their high days. I think, my experience is that most athletes will get better results that way.
One thing you mentioned briefly before is lactate. We know lactate is essentially half a sugar, and the body and essentially the mitochondria can reuse that as fuel as well. Now, if we talk about, exercise intensity domains: moderate, heavy, severe, extreme. And for those who are not familiar with those terms, I'd recommend going to watch a few videos. Maybe Mark Burnley's YouTube channel is a good place to start. He's got some great information on explaining those frameworks. One thing I'm really interested about is there a significant difference in the way the mitochondria is going to develop, whether we spend a lot of time in the moderate domain versus the heavy domain? And what are some of the adaptations that we might see regarding lactate shuttling and reuptake by the mitochondria in those different intensity domains?
Yeah, and like I said, I think we're actually writing a paper on this at the moment. I hope it won't be too far away, but yeah, those intensity domains are super confusing because if, there's different, the moderate domain is different from what most people would describe as moderate intensity. And then you've got zone 1, 2, 3 and 1, 2A and 2B and 1 through to 7 and things like that. So the hopefully not confusing anyone, but we've definitely seen that. So I guess, what we've how do I try and break it up? And I like your description before of like the and rather than the or. But if I was going to put into really broad distinction, so what we've seen is like the moderate intensity, so below the second lactate threshold, anywhere below there, and doing so. That's your longer, mostly continuous type exercise. I would argue that that's really good because you can get the high volume of exercise in there, good for increasing mitochondrial content, if that's what you're trying to do. We've also seen with the high intensity interval training that is also, you can get, because you've got the higher intensity, even though the duration tends to be shorter, you're still going to get a lot of total work there. So we also get really good increases in mitochondrial content in that area. And you may also see some a disproportional increase in some of the mitochondrial function or the mitochondrial respiration if we do that there. And then when we go up into the really high intensity, so sprint interval type training, for whatever, we don't understand why, but for whatever reason, that seems to be a really powerful increase in mitochondrial function. So we get disproportionately, compared to the increase in mitochondrial content, which is typically some there, but quite small, we'll get a much greater increase in mitochondrial function per mitochondria, if I can describe it that way. So that's kind of, it's probably a crude distinction, but that's the three that we've kind of focused on: low to moderate intensity, long duration. So classic sort of what people are seen on lots of blogs and stuff, I guess, typically sort of zone 2 type, people call that zone 2. High intensity interval training above the second lactate threshold and below VO2 max. We see you can get a high volume there, but also increases in content as well as function per mitochondria. But to get the greatest increases in mitochondrial function per mitochondria, then having some sprint interval training up there as well is beneficial for that aspect. And we're actually trying to do a little bit of research at the moment, because a lot of and you'd be aware of this from all your reading and work, but a lot of the research, for good reasons, we tend to focus on just one type of training. And so someone asked me a year or two ago, well, what would happen if you mix those all two together? So we're doing that at the moment to see whether, if you've got some low intensity, some high intensity interval training and some sprint interval training, if putting that into a periodized type training, can we get some of the benefits of from those individual components?
And that reminds me of I think it was a paper I saw a few months ago. It was in cycling. It was regarding the preseason, and I think that there was two groups. One that did only low intensity continuous training, and the other one included, and I forget if it was once a week or just spread out in different sessions throughout the week, some very short, intense sprints as well. And from memory, what they concluded is that those that had both the high volume of low intensity work, but also those little sprints kind of sprinkled in, seem to do better in the training in the following season. Whether it was potentiating their total mitochondrial pool and getting it ready for everything that was to come, because like you said, the demands of the low and the high intensity are quite different on the body, and maybe kind of hitting at those two ends of the spectrum, the really easy and also the really hard, but only for short periods of time, allows you to almost check all the boxes so that when you go back to regular training and competition and high intensity work, you're maybe a bit more ready for it.
Yeah, and I guess there's a little bit of debate about whether it's polarized or pyramidal, and it seems to be a little bit of semantics to me. But fundamentally, I think what you're saying there, I mean, there aren't. I'm not aware. I don't think any elite athletes do the same training all the time, just as they don't eat the same meal for breakfast, lunch, and dinner every day of the week. So, but yeah, it seems to be that getting a large chunk of your training, or this is for endurance athletes, but from that lower intensity, high volume, but also having some high intensity to very high intensity training. That seems to be what most athletes do. And I think from what you described there and from some of our research, in terms of hitting different physiological adaptations, I think it makes a lot of sense as well.
Do you see an important distinction in mitochondrial adaptation or development if we think about the first lactate threshold and we look below or then above that first lactate threshold? Is there some significant differences in those areas, or if we talk about mitochondrial development broadly, it's I guess the category of moderate and heavy domains kind of fused together is sufficient, or is there a distinction in the way that the body will and the cells will adapt to those different work intensities?
Yeah, it'd be interesting to do. We haven't looked at, and I haven't seen anything that specifically looked at that. I suspect, and it's going to be largely to do with the volume of training there. So, if you did high intensity interval training, just below the lactate threshold or just above the lactate threshold, I'm not sure you're going to, if you had the same amount of volume, I suspect you would get fairly similar adaptations. The only caveat I would add is that we've got some, I think, really cool research that we haven't published yet, but where we've looked at just in terms of changes, exercise-induced mRNA, and we had some people exercise just below the lactate, or the same people just below the lactate threshold and just above the lactate threshold. And there was even though it's not a big difference in terms of, if you think of watts on a bike, it wasn't massive, but there was quite a distinct difference in the signaling response. So there does seem, and we also, so jumping around a little bit there, but we also had another slightly lower exercise, and there wasn't much difference between the two exercises below the second lactate threshold. But as soon as we went above the lactate threshold, there's quite a distinct difference in the signaling response. So that suggests that there might be something important there. And if you think about, I guess one of the theories is that the metabolic changes or the metabolic stress with exercise is one of the drivers of the adaptive response. It would make sense that is an important exercise demarcator of an exercise intensity that might have different effects on the muscle and also the mitochondria.
Okay. One question that I've had for a while now in regards to modalities, training modalities, in developing that mitochondrial pool. Have you seen any research or data on the differences? For example, if we equate volume and that it would have to be maybe arbitrary work units. If we compare, for example, cycling and walking, or cycling and running, is there going to be? Have you seen any information on that topic? And the reason for my question is, and especially now, if we kind of jump back to the health side of things, I've worked with some physios and kines, and they used to have their patients walk a lot. And those are people that are usually overweight. Some of them had respiratory pathologies, so any small amount of work would feel really hard. And when they switched them from walking to cycling, even though it remained at a very low intensity, maybe 30 watts for most of them, they saw a significant difference in how they adapted to that exercise load. Are you aware of any research on comparing, I guess, the differences between walking and cycling, or running and cycling? And is there one that's quote unquote better than the other?
No, I haven't seen it. So do they find better adaptations with the cycling or with the running?
With the cycling, they really, when they switched from the running to the cycling, and did fairly close to the same amount of volume that they were doing walking, but now on a bike, within 3 to 4 weeks, they really started to feel a huge difference in their recovery and their total capacity, and just health markers in general. And I thought that was interesting.
Yeah, and it is that's why I asked, because some of the research we're looking at now, and I mean some of the stuff that George Brooks started doing 10 or 20 years ago. But the idea of lactate as a signaling molecule that might be driving, part of the driver of mitochondrial biogenesis. And so I think, one of the things that, compared to, with cycling, because you have that more, I guess, kind of consistent muscle, you don't get the same relaxation phase that you do get when you're running. So, and I think, even at a moderate intensity, I think a lot of people get more of that burning sensation in their legs with cycling rather than running. So it's interesting that the better adaptations, and I'm wondering if in that instance you're probably going to have a sort of having prolonged sort of lactate accumulation in the muscle with the cycling versus running. So it would actually, I don't have any papers and I haven't seen anyone specifically comparing that. And like you said, it's a really big challenge to equate the work of running with the work of cycling. But it would make sense to me that if you're able to have a greater exposure to lactate with the cycling, that might drive greater mitochondrial adaptations. And in effect, I guess that's what we were talking about a little bit before as well, is that we see when you go to above your second lactate threshold, there seems to be different signaling pathways being activated. And as I said, George Brooks's research, probably at least 20 years ago, showing even when you add lactate to muscle cells, that you get an increase in PGC1 alpha and other signaling molecules. So that's speculation, but it does make sense to me that anything that you can do to maybe promote some of this lactate accumulation may drive some of these additional mitochondrial adaptations.
That's really interesting. I definitely heard about lactate as a signaling molecule, in part linked to what happens with fat stores breakdown and triglyceride availability, in terms of substrate, as in when lactate starts accumulating, we downregulate our fat oxidation. But I hadn't heard it linked to, like you said, the PGC1 alpha, which is, if I'm correct me if I'm wrong, but one of the markers of mitochondrial biogenesis. So essentially the creation of more and more mitochondria. Is that correct?
Yeah, exactly. And there's a paper, if people are looking up, so the first author was Hashimoto. So I can't remember, I think it was 97 or something like that. So good 20 years ago. But there's a Japanese group as well that have done some studies where they've injected lactate into the muscle of rodents and seen the activation of these mitochondrial biogenesis signaling pathways and things like that. So I think it's it's becoming, yeah, when most people think of sort of lactic acid as a fatigue sort of agent, but as you mentioned, lactate as a substrate that the mitochondria use, but also increasing evidence that this idea that I guess the lactate being produced is like an energetic stress signal that communicates to the muscle to drive some of these mitochondrial adaptations.
That's really interesting, and that does change my framework of reference a little bit, in the sense that you mentioned the zone two and the low intensity, and what I would call zone two, because again everybody's got their own definitions, would be
Yeah.
below the first lactate threshold. So you would be in zone if we had to equate it to fat max, for example, where you would have your maximum fat oxidation, which would usually correspond to minimal lactate values on a lactate step test, for example. And this is obviously a very low stress type of intensity where you don't even have any lactate accumulating in the blood. And then once you pass that transition from, say, zone 2 to zone 3, or that first lactate threshold, and you start to see slightly elevated but stable lactate in the blood. And I've seen some really good results combining those two, and having, for example, CrossFit athletes, or even, like I said, team sport athletes, soccer players and rugby players, do both. So do some zone 2, so very easy work, where if you were to measure lactate during exercise, it would be at its lowest levels. And then also do some zone 3, zone 4 work, so kind of between those two thresholds. So it's hard-ish work. It's, say, 5 to 7 out of 10 maybe on the RP scale.
Yeah.
And combining those two types of work, bringing some really good results. And it did seem like that zone 3, that kind of medium but still pushing a little bit intensity, was very productive work. So I'm wondering now if actually most of the adaptations came from that work, and the zone 2 really easy work was more the support structure in terms of, low intensity and then just broad volume, but without any stress added to the body. And that's really interesting that you mentioned that distinction and that role of lactate and signaling.
Yeah, and with some of the work, we just finished a study where we did a lot of training. So I think you're zone 2, but just below the first lactate
Yeah.
threshold. And we did a lot of volumes. I think they were doing 90 to 120 minutes, 4 days a week. And probably, yeah, some of the changes in mitochondrial volume were a little bit less than I would have expected, to be honest, in terms of the total volume. So we want to do some follow-ups, but my thinking is probably getting above the first lactate threshold is probably going to be important for that low to moderate intensity type activity, for that volume work.
That's interesting. And I guess then for me the question was always of how to, how much volume can you handle, because obviously once you pass that first threshold, you have elevated lactate in the blood. We also see that usually corresponds to catecholamines and other stress hormones and markers going up. So or from that, I assume that this work is a little more stressful than the zone 2 below first threshold work. So I guess you still should push that volume of between threshold work, but you just have to make sure you can recover from it. And maybe that's where the lower intensity zone 2 stuff comes in and allows you to recover better from that elevated, where it's not high intensity, but it's more than that zone 2 type of intensity.
No, exactly. And I think that's where I always have to be, careful with some of these conversations. I guess I'm talking specifically about mitochondrial adaptations.
Mm-hmm.
And obviously, we're talking about mitochondria, and as important as we might think they are, but there's other considerations in terms of a training program. So yeah, I think you're spot on. There's definitely a role for that, even lower intensity training. But in terms of we haven't done this experiment, but I suspect that if we did the same volume of training above the first lactate threshold compared to below the first lactate threshold, my hypothesis would be that you'd get better improvements in mitochondrial content with the same amount of work just above the the first lactate threshold.
That's excellent. I'm really excited that you blew up my framework on that. So I'm gonna have to work on this in the coming months. Um, let's stay on the health side. You talked about mitochondria's role in mitigating reactive oxygen species production. Can we go in that direction for a little bit?
Yeah, I guess, I mean, so the reactive oxygen species is coming, not just from the mitochondria, but also from the muscle. And again, at the caveat, I won't claim to be an expert on reactive oxygen species, but obviously they can cause damage to the lipid membranes, not just in the mitochondria, but in other cells inside the muscle. But you're going to get an increase in reactive oxygen species as you're exercising and as your mitochondria are being, I guess, stimulated to produce more energy. But with that will drive adaptations so that for the rest of the time when your mitochondria when you aren't exercising, that so more the basal type state, so the extra 22, 23 hours that you're not exercising, the mitochondria will be producing less reactive oxygen species. So that's part of one of the adaptations with training and with the mitochondrial adaptations is that the mitochondria become less efficient, and as part of the electron transport process, more of that is being directed producing energy and less of that is going to be going to produce reactive oxygen species.
Is that one of the reasons that exercise is so beneficial to health and just physical activity in general, in your opinion?
I think it's probably part of it. And I think it's an interesting thought, because I guess people always, there's always cool articles and discussions about exercise mimetics. And so, there's probably some sort of vitamin or pharmaceutical thing that you can do to prevent the reduce reactive oxygen species. But you've got everything else that's going on with exercise as well. So, I mean, to answer your question, I think it's probably a small part of the benefit, but there's lots of other benefits from exercise that are going to be contributing to the health benefits of exercise.
Another question in that realm is, and I don't know if you have any information on that, is the role of mitochondria in the production of melatonin. My understanding is that, or something I've come across recently, and I don't know how valid it is that we often think of melatonin as maybe a supplement we can take to help us go to sleep at night, and we know that it's something that is also produced by our pituitary gland during the night. And one thing I came across is that this might be kind of a backup mechanism, whereas during the day when there's light outside and our skin is exposed to light, mitochondria is the main producer of melatonin in the body, melatonin being a very high antioxidant, if I'm not mistaken. And then it's essentially our brain that takes up that work during the night when we don't have light exposure. Do you have any information related to that?
I haven't done any work on that, and and this is my superficial understanding, is I think it's almost, your description there is exactly as I understand it. But I think from the melatonin that is being produced by the brain is mostly the circulating melatonin. So I think that's when you talk about, the circadian rhythm, and as you said, the fluctuations in melatonin, and that help to, prime you to go to sleep and all that sort of stuff. You're right, though, the mitochondria also produce and use melatonin, and it does have an antioxidant role in the mitochondria. But my understanding is that melatonin isn't released into the circulation. It's being produced and then synthesized and also used within the actual mitochondria. So it's kind of like two different pools of melatonin if
Mhm.
that makes sense. And within, helping to prevent damage to the mitochondria, I think the melatonin that is produced by the mitochondria does have a role
Mhm.
there. But my understanding is that that can be transported out of the mitochondria, but it doesn't that's not contributing to the circulatory melatonin pool.
Okay. If we talk about substrate utilization, so to make it simple, we're gonna primarily use sugars and fats in our mitochondria to recycle that ATP. Um, can you talk about the differences in how an organism or someone will handle carbohydrates? If we think of high level athletes, they're likely going to be consuming a lot of them, and because they have the infrastructure. But on the other hand, and that's another question of Vincent, if you look at sedentary people, they lack mitochondria, and it seems like carbohydrates have the opposite effect that carbohydrate has on high level athletes, whereas it's not as beneficial. Can you talk about this a little bit?
I think so. Yeah. So I think as you alluded to there, I mean, there's two main fuel sources that that the body will be using. So using the stored and circulating fats, and then also the carbohydrates. And there's nearly always going to be a mix of those two. So another nice example of your and/or analogy from the beginning. When people are doing, and anybody, low intensity activities, sleeping, things like that, most of the energy that's being used is going to be from the oxidation of fats by the mitochondria. As you increase in intensity, then you're going to increasingly be adding carbohydrates to the mix, until once you get up to very high intensity exercise, it's going to be predominantly carbohydrates. In terms of the usage of those different substrates, the main thing that will change with training is I guess the absolute intensity that will kick in
Mhm.
when you start to change that distribution. So really, really early on, untrained sedentary type people, quite early on, they will have to start increasingly including carbohydrates into the energy mix to be able to supply the energy that they need. Whereas with endurance training, you're pushing that towards the right. So it's going to be a higher intensity where you can predominantly using fats to supply energy, and you can delay the usage of the carbohydrates. So I guess that helps to protect the limited glycogen carbohydrate pool that is in the skeletal muscles.
And yeah, if we bounce back to the performance side, I guess that's one of the upsides of building that mitochondrial capacity, is that you are going to spare your glycogen stores until for when you really need them. And it's probably some of what we saw on the tour with Pogacar, with the fact that he was flat, he was on empty, and he just couldn't, I guess, couldn't assimilate the food anymore to try and prop those glycogen stores back up. And if you don't have those, then you're not going to be able to rival with the best. And I think that, I guess, that glycogen sparing mechanism is a really important one if we think about performance and building that mitochondrial base.
Yeah, exactly. And so I think that's, part of what's, obviously there's lots of different adaptations, but that's one of the adaptations there, is trying to have this glycogen sparing capacity by having mitochondria that are better able to utilize, the ample supplies of, fats that are running around there.
Mm-hmm.
And it's not only a quantity that's being used, but so the reason that you need to add in the carbohydrates is because using fats, they won't be able to supply energy as quickly as it's needed. So that's also part of the adaptation that will occur with the mitochondria that'll allow people to exercise at a higher intensity before they need to tap into their carbohydrates to be able to supply their ATP energy producing needs.
That makes sense. Another question from Vincent, and Vincent is doing some really great work in the markers of aging. So we're back on the health side. His question goes as follows: we observe a deficiency in coenzyme Q10 with age. This deficiency can be up to 90% in certain tissues, such as the pancreas and the heart. Can training, low intensity, medium intensity training, reduce these percentages, or not?
Yeah, I have no idea. So I haven't looked into that itself. I'm trying to think when we just done some work on the proteomics. So I mean, with exercise, we see basically an increase in all. You know, we're not able to measure all of those 1,200, 1,300 mitochondrial proteins, but we do see basically all the ones that we do measure increase. So I haven't had specifically looked for coenzyme Q10, but I suspect that it would make sense that the amount of that protein is increased with training. So whether that can help with some of the, I guess, the anti-aging or protective effects of exercise, that's a possibility, but it hasn't been something that I've specifically looked into.
Staying on the topic of health and talking about internal organs, have you seen in your work? I know you've focused a lot on muscle, but is there I don't know, I wouldn't call that a transfer, but do we also see an increase in mitochondria in internal organs as a result of endurance training?
Yeah, it's a really cool question. It's a bit, yeah. We mostly do human studies in my lab, but it's easy, relatively easy to get a muscle biopsy, but getting other organs is a little bit more challenging. But we've done over the years just a couple of animal studies, and maybe it's not surprising, but we do see increases in mitochondrial function in other tissues other than the skeletal muscle. So we haven't published this, but I think we did liver and kidney and things like those. And so it's interesting because obviously, your kidneys and liver aren't contracting when you're doing exercise, but whether it's blood flow or whether it's the idea of having these exosomes that when you do exercise you're communicating with the other organs. But we've definitely seen that when you do exercise, or when the animals are doing such treadmill running, when they do exercise, you're also seeing increases in mitochondrial respiration in these other organs and not just the muscle.
So that would, I guess, support the idea of physical activity in support of global health, not just the muscles, but the body as a whole and all its components.
Yeah, absolutely. And I think that's where, again, sort of circling back to one of our early discussions, but yeah, it's interesting, you're doing exercise and it seems to be improving mitochondrial content and function. It's been linked with Alzheimer's and Parkinson's and things like that, which presumably, don't have a lot to do with the skeletal muscles. So with the exercise, we're, broadly seeing health effects in most of the organs that have been measured, not just the skeletal muscle, and that also includes mitochondria. So doing exercise develops your mitochondria in tissues, in other tissues, not just skeletal muscle, but also in other tissues.
You talked about the importance of health, obviously, and that you're starting to think more in that realm. Obviously, still looking at performance, but seeing what we can gain from those findings in the health realm. Where would you like to see research and practice when it comes to training and health go in the coming years?
I think we were just talking about this other day. I think in terms of you see like the classic in terms of diagrams in terms of bone mass and osteoporosis, in that yeah, it's quite hard to you. It seems like the decline is inevitable,
Hmm.
no matter almost what you do. And so I guess part of the goal is to, you know, money in the bank, get your bone density as high as possible when you're 20, 30, 40 years old. And so then you're falling. Yeah, you can probably slow it with exercise. And I'm not sure you can prevent the decline completely, but yeah, getting to a high value when, you know, between 20 and 40, so that you've got, if there is a fall, you know, you've got a buffer to prevent that. So I think that that's also, I've been thinking that in terms of mitochondria as well. You know, what do, you know, almost where do we need to be at when we're in our prime, so that, you know, if you can fortunate enough to hit 90 or 100, that you can still be in a in a good place there. And so, and I saw I think it was a tweet, I can't remember, from someone, you know, suggesting that you know, to keep your VO2 max above 50, you need to do 10 hours of exercise per week,
Hmm.
which I think, you know, most people probably, you know, a lot of, you know, think of 50, 60, 70-year-olds would, you know, or we know from the epidemiological stuff that, you know, most people aren't getting the 150 minutes. So
Hmm.
to get that, so I think that slightly a rambling answer, but just that concept of, you know, I think, you know, sometimes, you know, lifetime exercise is great, but where do we need to be trying to get people at, so that they're going to be in a good place to be able to deal with the inevitable decline that's probably going to happen with aging. So I think that's particularly interesting. I think also, you know, maintenance doses, we don't have a lot of information on that. And I think, you know, we've got some pretty good information on what you need to do to increase your mitochondria,
Hmm.
but, you know, inevitably there can be periods in people's lives where they're going to struggle to get the exercise that they need. So what is you know, if there is a reason that you can't get the you know, the recommended amount of exercise, what is it, how much exercise do we need to do to be trying to, and not only how much but intensity, etc., to try and maintain our mitochondria? I think that's a pretty interesting question as well. I think in more of a scientific type perspective, you asked this right at the beginning, is that what exactly is going on to improve mitochondrial function? And I don't think we really know that. So that's something that's occupying my thoughts lately as well.
That's really interesting. And back to what you said regarding bone density, I think of strength in that realm. And we I remember seeing those numbers years back, and seeing a strong inverse correlation between you talked about VO2 max, which could be a marker of aerobic fitness, and I think they were talking about strength, and which is going to be linked to bone density as well. If you do resistance training, your bone density is likely going to be higher, and those two factors together had a strong inverse relationship with all cause mortality. So essentially, if you have a higher VO2 max and you're stronger, you're going to live longer and better, which is essentially what you were getting at there.
Yeah, and I think also, yeah, and I think that's, it's interesting, some of the work we've been doing, that a lot of the, yeah, a lot of the public, or nearly all the public health messages are around 150 minutes of, you know, moderate to vigorous intensity exercise per week. But there's very, and most of them kind of have, it'd be nice if you could do a little bit of flexibility and strength work, you know. But I think that's going to be a bit of a, you know, I think you're going to see a bit more of a move towards promoting the strength training as well. And like you said, some of the data I've seen, like the links between, you know, being able to, I don't know, you know, squat strength or whatever, and quality and quantity of life are pretty impressive. So I think there's going to be a more of an emphasis on that, and maybe even mixing the two, because if you're trying to maintain muscle mass and synthesize proteins, you need lots of energy. And so there's I think there's quite a few researchers doing some interesting work on that. Is, what is the link between mitochondria and muscle mass as well? And so I think that declining mitochondria will also contribute to declining muscle mass and sarcopenia, because you don't have the energy that's needed to be able to maintain your muscle mass.
So it's again a question of and not a question of or. You need to probably work on both at the same time. How do you reconcile the 150 minutes, which is about 2.5 hours, and the 10 hours that you mentioned before? That's a factor of 4 in terms of the difference in weekly prescription of exercise. Where do you fall on that 2.5 to 10 hours per week
Yeah,
in terms
I
of
mean,
recommendation?
yeah, and I think it's interesting, and that's not my, I'm not involved in a lot of those discussions, but I think in the terms of the public health realm, there's there's kind of two questions. One is, what would be ideal, and what have we got a remote chance of getting people to be able to do? And so I suspect that, if you wanted to give the optimal health message, you're probably looking closer to the 1 to 60 to 90 minutes per day. But most people aren't, if you look at the data, most people aren't doing 150 minutes per day. So, 150 minutes per week, sorry. So, a recommendation to do 5 or 10 hours a week, it's not going to happen. So I think when you look at public health messages, it's really about what's, given where people are at the moment, what is an achievable goal. And so I think 150. If most people aren't doing 150 minutes, so if you can do that, that's great. But if you're someone who's already doing exercise and you're trying to think about how you can, get the best health benefits, I think that's probably going to be closer to 60 to 90 minutes a day.
That's good, because people listening to this will probably want to know what's the best, and what do they need to do in order to get the most and probably optimize that health. And also, obviously, like you said, it's interesting that your perspective on an optimal volume for health is around 10 hours a week, and a lot of maybe competitive athletes don't even train that much per week, so they would probably benefit from increasing their training volume. And an easy way to do that is to add some easy work in there, and to get some more minutes just moving. And at its base, I often go back to what were we built for as humans, and how have we lived on this planet for thousands and hundreds of thousands of years, and that's mainly moving around. And obviously we've spent the last hour and a half not moving, sitting in front of a computer, so it's not a great start of the day for me, or end of the day for you. But it's I guess just the physical activity factor in our overall health and our makeup as human beings has to be part of the conversation as well, right?
Sometimes, in terms of, this is now getting a little bit away from the mitochondrial talk, but in terms of public health, that quite often people want to know, what's the optimal exercise, what time of day should I do it, what intensity, and quite often then the next question I'm asking is, well, what are you doing? And they're going, oh, I'm not doing anything. And I'm going, well, okay, how about we start with doing something and doing something every day, and then that's when we can start thinking about, after once a habit has been formed, then you can start tinkering with exactly what needs to be done there. And so that's where I think the 150 minutes a week is a good goal for a lot of people. The US and Australia, I think, are pretty similar, that can't remember, probably like 50, 60% of people aren't even hitting that goal. Once you're hitting, 150 minutes per week, doing, 20, 30 minutes on most days, then I think you can start having the conversation about, what type of exercise to add in, and, then maybe you can start thinking about time of day or intensity and things like that. But, I think the first step is doing something, and then the second step is tweaking it to make it as effective as possible.
Fantastic. Well, David, I really want to thank you again for doing this podcast. It was an absolute honor and pleasure for me to be able to pick your brain for an hour and a half. Do you have any final thoughts, closing thoughts for the audience of this talk today?
No, great. I think it's been great to talk to you. Thanks to Vincent for his tough questions. I think just always add the caveat. I mean, a lot of our research is about the mitochondria, which are obviously important. And as we touched on earlier, there's obviously other considerations. As much as I sometimes don't want to admit it, there are other things that are as important or more important than mitochondria. So I think it's an important consideration, but there's going to be other factors that are going to be also important when it comes to getting whatever each person wants to get out of their training program.
Fantastic. David, if people want to follow your work, where can they find you online?
Yeah, they can normally, Twitter's quite a good, I think it's still a good place. But my Twitter handle is @BlueSpotScience, so they can go on there. If they look me up, they'll find me on the internet if they want to, find any papers. But I've been a bit slack lately. But normally, if we've got new research, or if I see new research, especially related to exercise training and mitochondria, then I'll normally tweet that. So that's probably a good place to start.
Great. I'll put your Twitter link in the description of the podcast. Thanks again, and have a great rest of your day.
Have a good day, mate.
Thanks for listening to this episode. If you would like to support the podcast, please take a few moments to leave a written review and a 5-star rating on Apple Podcasts. If you want to watch this episode again, you can find the full video recording on my YouTube channel. You'll also find hundreds of hours of free content, all my podcasts, my thoughts of the day, structured presentations, and more. So don't wait, go subscribe, and I'll see you in the next episode. Take care.